last but certainly not least is proteins. These do a lot of things and they have complicated structure, so this video is going to be longer than some of the others. So the monomer for proteins is an amino acid, and then the polymer is a polypeptide, which we will sometimes call a protein. And it gets a little confusing whether you're talking about a polypeptide or a protein. I will explain the difference. The distinction between the two is not all that important for us. So the amino acids are held together by what are called peptide bonds. And there's this word here which I just wanted to introduce. If you have a long strand of amino acids, it is called a polypeptide. If you have 2 or 3, you might call it a dipeptide or a tripeptide. If you have a short strand, 10 to 20, then we call it an oligopeptide. So it's too long to mention it as a tri or a bi or a quadrupeptide, but it's too short to be considered a true polypeptide. So it's just this weird oligopeptide word that I want to introduce. We will start by talking about what amino acids look like. So you want to recognize this structure as an amino acid. They all have the same basic structure. There is a central carbon in the middle. And then each amino acid has an amino group on one side, a carboxylic acid group on the other side. So this is where they get their name amino acid, because this hydrogen can be released as a hydrogen ion, decreasing the pH of a solution. And then what makes each amino acid different is what we call the R group. The fourth constituent or substitute, the fourth thing that is connected to the carbon. So they're all going to have the amino, the carboxylic acid, and a hydrogen. Then the fourth bond partner is what makes each amino acid different. And it is the side groups on each amino acid that give that amino acid its physical and chemical properties, and it's the collective properties of all the amino acids, or all the amino acid side groups in a long chain that gives the protein its final shape and function, or physical and chemical properties. So just to give you a couple as an example, this is glycine. Glycine's R group is just a hydrogen, so it's very small. So you tend to find glycine in places where the protein backbone curves, because this hydrogen can fit into a little curve like that. With aspartic acid, this is an example of an amino acid side group that is still acidic when incorporated into a peptide chain. So we haven't gone over how the peptide bond works yet, but this acid part goes away when the two amino acids are bound to each other. So in the peptide form, when they're bound as part of a polypeptide, most amino acids do not have this hydrogen, or they all don't, so they can't function as acids. But this amino acid has this carboxylic acid group that hangs off into space when it's incorporated into a protein, and it can let go of that hydrogen ion and act as an acid. Some amino acid side groups have an amine group, this NH2. It can become, as we've seen before, that's supposed to be a 3, and pick up a hydrogen ion. So it can function as a base by decreasing the number of hydrogen ions in the solution, thereby increasing the pH. And then cysteine here is really special. It has this sulfur group in it, which can form special bonds with other amino acids in the chain called disulfide bridges, which is important for the ultimate structure of a protein. And we'll get to that later if I have a picture of a good example of that. So here is just to show you what the peptide bond looks like. So it is also, if you are putting together amino acids to make a protein, this is dehydration synthesis, right? So you pull the OH off here, you pull the H off here. No, the OH, I'm messing it up. You pull the OH off here and the H off here, and you make water. And then now this carbon is directly attached to that nitrogen there. And because it's carbon to nitrogen, as opposed to carbon-carbon, like say this bond here, it is special or different than other bonds, so we call it a peptide bond. So then, now we need to talk about the different levels of structure. And I will review by saying you need to be able to recognize an amino acid when you see one. Excuse me. You will need to be able to recognize a fully folded protein when you see one. But I will not give you visual examples of primary, secondary, tertiary, or quaternary structure. You just need to know what they are. So don't worry about these pictures. But here we go. And then we'll also talk about conformation. I just wanted to make sure that shows up in one of the next slides. So the primary structure of a protein is simply the sequence of amino acids. You don't even need a picture. You could just read it out. Aspartate, glutamate, lysine, cysteine, whatever. It's just the amino acids as they're strung together in the peptide chain. Secondary structure is what we call localized structure. So if you have a larger protein, there might be parts of it where the polypeptide chain is wound up into little spirals like this alpha helix or this beta pleated sheet here. And I don't really have time to go into like the chemistry or the actual mechanics of how this works. But just know that alpha helixes and beta pleated sheets are examples of secondary structure. This will become clearer when we see what tertiary structure looks like. So this is the final shape that one polypeptide takes. So it is as it says here, a single strand of amino acids or a single polypeptide. So there's one end here. I don't know if that's the carbon or the nitrogen. I forget what the convention is. And then this other end here. But one of these is going to be a nitrogen end and one of these is going to be the carboxylic acid end. And as you follow the protein backbone, you can see here's an alpha helix, so some local structure. This arrow is supposed to be a beta pleated sheet. So you have a couple of beta pleated sheets and a couple of alpha helixes. That's the secondary structure. All of that is combined with these parts here are called random coil, where they don't have like a set structure. But that final shape then is called the tertiary structure. So if you see something that looks like this and it's just this, you could call that a protein. So if a single strand of polypeptide performs the function that the body needs for it to perform, then we call that tertiary structure the mature protein. In some cases, in order for functionality to happen, there have to be multiple polypeptide chains stuck together to make the final functional shape. In that case, we say this protein has quaternary structure. So the mature protein contains 2 or more polypeptide chains. So we could say a protein is 1 or more polypeptides that, when properly folded, perform the function for the body. So this happens to be hemoglobin, which has 1, 2, 3, 4 different peptide chains, and you can see up here they're all different colors. This final shape then, whether we're talking about the final tertiary structure or the final quaternary structure, the shape that a protein takes when it is functional is called its conformation. So that's an important word that we want to remember. So then we need to go over protein function. This is where things get a little complicated because proteins are like metal, right? You can use metal to make lots of different things. It can be a machine, it can be structural. Metal can also be used to make tools, right? So it's all sorts of different things. Structural examples of protein is in your hair, and you have proteins that reinforce the concrete-like mineral matrix in your bones to give it some flexibility. Proteins can act as motors, like they actually move stuff around. So it is motor protein in your muscles that pull on other proteins and make your muscles get shorter. They can act like receptors. If I go back to this picture here, these are all proteins. That's a protein, that's a protein. This protein right here has one end that is inside the cell and another end that sticks outside the cell. It can interact with things outside the cell, which is going to cause it to change its shape, which then causes stuff to happen inside the cell. So that's an example of a protein acting as a receptor. So it's sort of like, right, the button on your keyboard. You push the button on the keyboard, and that's outside the computer, but then something happens on your computer screen. That's what receptors do for your cells. But one of the most important things that they do, which we're going to take some time to talk about now, is they act as enzymes. And I think we already covered the term catalyst. I'm trying to remember where we are in the chapter because I'm just randomly making videos. Well, I won't go over it maybe. Yeah, I'll just write it in case we haven't covered it, but I'm pretty sure we've covered it, right? Enzymes are catalysts. And a catalyst is anything that speeds up a chemical reaction. So it makes it easier for the reaction to happen at room temperature so that the reactions happen faster at room temperature. And the other important thing to remember is that a catalyst is not altered or consumed during the reaction. So an enzyme can catalyze a reaction, like let's say the formation of a peptide bond. Right, so there's an enzyme that is responsible for making sure that this bond happens when and where it needs to happen, and it can do it multiple times. So it'll link these two peptides or these two amino acids together, and then it'll put another amino acid here and another amino acid there. So the same protein enzyme can catalyze thousands of peptide bonds to string together amino acids to make a polypeptide. So enzymes are hugely important to how your body works. They regulate almost all of the chemistry within your body and make sure that it happens at the right time and in the right place. The way enzymes work is by decreasing what is called the activation energy that is required to get a reaction to happen. So even if the product is at a lower energy state than the reactants, meaning this reaction gives off energy, you have to put some energy into it to start it. So it's sort of like a fire, right? Once a fire gets going, the energy released from the fire causes fuel next to it to burn. But there has to be a spark or a match to start the fire. That's what enzymes do, is they get reactions started. So like if we go back to the protein, right, before you can get this peptide bond to form, you have to break this bond here and that bond there. So that is the activation energy required to form a peptide bond, and the ribosome makes it easier for those bonds to break, and so that you can form this new bond. So again, here, this is your activation energy. That's how much energy is required to break the old bonds so that you can form the new bonds. And Enzymes just make that less. So the less energy there is required to start a reaction, the more likely it's going to happen at any given temperature. So the way enzymes work is we say that the substances that the enzyme is going to catalyze, whether it's taking two things and putting them together or taking one thing and taking them apart, the thing that the enzyme acts on is called the substrate, and then what you get from the enzymatic reaction is a product, but you notice you also get the enzyme back. And then the other important word to cover here is active site, which I will illustrate in the next picture. That is the part of the enzyme that interacts with, oh, this says products, but it should be substrates. Oh my God. Eric, what is wrong with you? So the active site interacts with the substrates. So here you could imagine that these are your two amino acids that you're going to stick together, or maybe this is glucose and fructose, and you're going to make, no, that's not fructose. Yeah, sucrose. There would be an enzyme that binds to the glucose, binds to the fructose, encourages them to break their bonds, remove that OH and the H, so that the new bond can form. And then once you have the product will leave the enzyme. So this part right here is called the active site. Yeah, so we've covered all that. This is just a picture from a poster that whoever posted it said it's Creative Commons, but I might actually be violating copyright here, because I'm pretty sure they didn't get permission from the publisher of the poster, but who cares? So if you look here, each one of these blue things that are written in blue, maybe use something besides blue to highlight it, right? There's some blue writing, there's some blue writing, there's blue, there's blue, there's blue, there's blue. Everywhere you see blue, that is an enzyme that is catalyzing each step of the reaction, right? So this becomes that, which becomes that, which becomes that, which becomes that. And then if you have this enzyme, this happens. If you have that enzyme, this happens. So they're directing all of the chemistry. And if you look at the totality of the biochemistry that we know goes on in your cells, there is a whole lot of blue here. So the enzymes really do regulate all of the reactions, and they make sure that they all happen when they're supposed to and where they're supposed to. Because if you don't have an enzyme, then the reaction's not going to take place. And we'll learn later that your cells can actually control where in the cell any protein is. So if you put all the proteins that perform a certain chemical reaction in one little bubble, then you can control where the chemistry takes place. Excuse me. So just examples of some of the things that enzymes do, right? So they synthesize all of your hormones. As we just saw, this picture right here looks like cholesterol being turned into different things. So this is all your cholesterol biochemistry. So whether or not cholesterol gets turned into estrogen, progesterone, aldosterone, whatever, depends on which enzymes are acting on it. They make all of your neurotransmitters and other chemicals that you need within the body. It is enzymes that build up all of your macromolecules. So whatever the ribosomes that read RNA and synthesize your proteins, that's an enzymatic reaction. Your DNA is made from enzymes, and it is processed by enzymes. Sugars are converted into fat and vice versa by enzymes. Anything that gets converted into ATP, that is done by enzymes. And as I just said, copying DNA, protein synthesis, all of this is controlled by enzymes. Let's see. Oh, this is... Well, we'll go over this. I don't want to get into too much detail about how enzymes work because this is not a biochemistry class. But because they are powerful and they do a lot, your cells exert a certain amount of control over enzymes. The easiest way to regulate whether an enzyme is functioning or not, or how it's functioning, is to simply regulate how much of that enzyme you are making. So how much protein synthesis of that enzyme takes place. And cells can also degrade proteins when they are done with them, so that the enzyme is no longer catalyzing the reaction if you no longer need it to. Enzymes also have on-off switches, and quite often it is the addition or removal of a phosphate group that is going to turn an enzyme on or turn an enzyme off. So in the way you want to think of it, the analogy I use is that phosphate groups, it's this PO4, and one of them has a minus on it, and there might be something else here. Oh, it's another oxygen. All right. This is a relatively large thing to add to a protein, and it just changes the way all of the amino acids near this phosphate interact with each other. So adding a phosphate is sort of like adding 2 or 3 new people to an already crowded dinner table. It requires a lot of moving around and shuffling of things. So when you add or remove a phosphate from an enzyme, it greatly changes the shape of the enzyme, especially the shape of the active site, and that can then be used to turn an enzyme on and off, so that they're always around when you need them. And then all you have to do is add or remove phosphates, which is really quick, right? Much quicker than synthesizing a whole protein or degrading a protein. So this is how your cells, in large part, control whether enzymes are active or inactive. Then the last thing we need to talk about is protein denaturation, which means it loses its conformation. If an enzyme loses its conformation, that is going to disrupt the active site, so it is no longer going to work. But this also applies to just structural proteins. None of the proteins perform their function properly if they don't have the proper shape. So remember, before we talked about structure and function complementarity. That applies especially to proteins. So they are tools, they're machines, right? And if you have the wrong screwdriver, it's the wrong shape, it's not going to work. If you don't get the right size wrench, it's not going to work. You can't use a hammer when you're supposed to have a chisel or something. So shape really matters when it comes to proteins. They lose their shape, they lose their function. If you expose a protein to heat, that's not supposed to be there, it will lose its shape. And so what happens if you apply heat is that your heat is molecules moving. And so you're making all of the parts of the protein start to vibrate and move around a little bit. And if they vibrate and move too much, they just fall apart. Like they can't hold all of that energy and still stay in one shape. Right? They're like children after lunch. They're all full of sugar and they can't sit still in the classroom, so nothing gets done. Same thing with the proteins. And then pH can also change their shape. I will talk briefly about how this works, but I don't expect you to necessarily understand it. But you will recall that we have basic amino acid side groups. Sorry, this one is basic. And acidic amino acid side groups. Here they are drawn without their charges. So let's just look at this one in particular. And assume we have a protein here like this that's got all of these NH groups on it. So NH2 is going to be hydrophobic. It's not going to bond with water. So on a protein with that amino acid, the NH2 groups are all going to be in the middle, right? Because the water molecules are all out here. And the water molecules need somebody to hydrogen bond with. And NH2 can't hydrogen bond, so they all get stuck towards the middle. But if you dump this protein in acid, all of these NH2s then end up becoming NH3s, right? Because you've added a whole bunch of hydrogen ions. And now they have a positive charge. This protein is then going to unfold so that these NH3s are now on the surface and can hydrogen bond. So this is why if you put proteins into solutions that are the wrong pH, it changes the charge distribution on the acidic and basic amino acid side groups, and then can turn hydrophobic amino acids hydrophilic, and it can turn hydrophilic amino acids hydrophobic, and then the protein just unfolds. So that is like if you've ever left chicken in a marinade, like a lemon garlic marinade overnight, and the edges or the very thin pieces of the chicken turn white, that's the acid denaturing the proteins. You have essentially pre-cooked part of the chicken. That's also how ceviche is made. If you've never had it, I highly recommend it. I'm not really a seafood person, but ceviche is delicious. You take small pieces of light, delicate seafood, scallops, white meat. Some people use shrimp, but I think they cheat by pre-cooking the shrimp. And then you just leave it to sit in lemon juice with garlic and spices, and the lemon juice, or maybe it's lime juice, doesn't matter, cooks the fish, and it comes out, you know, acidic, delicious, cooked, delicate, cold fish dish. It's really good. It's one of the few fish dishes I like. You guys don't care, but I'll shut up now. We're done with proteins. That was 30 minutes. Thank you for hanging in this long.